Everyone, welcome to lecture 4 of BMS 300. Today we're going to talk about energy metabolism. And before we get into the overview of what I'll cover today, I want to just begin with this slide, which I hope you're all familiar with. You remember when we talked about it? When we went through the slide? Day 1, yes, thank you. I would have been very sad if no one responded. Yeah, day 1, I talked about how the entire course is summarized on this slide, right? And I want to keep coming back to the slide so that you're aware of where we are and how we got here. So we started out lecture 2 by talking about the structure of water, because that is the major component of cell. That led us to the discussion of how various substances can interact with water. So you have hydrophobic and hydrophilic interactions, and we also looked at amphipathic interactions. And when you consider the cell membrane, it's made up of these phospholipids, which are amphipathic structures, and the cell membrane also has a lot of these proteins which are embedded in it, and that led to our discussion of the structural complexity of proteins. You have primary, secondary, tertiary and quaternary structure, and that structure is essential to inform what sort of function the protein has. And so today we're going to talk about how ATP is required by the cell to make proteins, and is also required by the cell for pretty much every single function which is required. So anything which requires energy is going to use ATP, because ATP is the energy currency which our cells can recognize. So with that overview, let's see what are the topics we'll be discussing today. So the few questions which I hope you can answer at the end of metabolism is answering the question, how does the body extract energy from food? So when you think about the macronutrients we eat carbohydrates, and the carbohydrates can be broken down by both anaerobic and aerobic pathways to make ATP. And when you think about fats, this beta oxidation step is unique to fat breakdown, and then the other steps which are involved may be common to both the breakdown of fats and carbs as well. And finally, when you think about proteins, proteins will undergo deamination, so that is a step which is unique to protein metabolism, and then the remaining steps for protein breakdown would be common to carbohydrates, proteins and fats, right? So because the carbohydrate metabolism has steps which are common to all three, we're going to start with that, then move to fat metabolism, and finally to protein metabolism. And then I want to talk about the second question: what is the connection between metabolism and respiration, right? So we see that metabolism is linked to the functioning of the digestive system, respiration is linked to the functioning of the respiratory system, but my goal is to tell you at a cellular and molecular level how they are integrated, right, in today's lecture. When we move on to unit 4, we'll see how this integration at the cellular and molecular level will translate to the whole organ system. So this is one of the learning outcomes I have for you, to be able to integrate what's happening at the cellular and molecular level all the way to the organ level. So in simple terms, I'll explain why we breathe in oxygen and why our cells produce carbon dioxide at the cellular level. And we'll finally be talking about what fuels our body uses for different types of activities, right? So that's the overview of what we'll cover in metabolism. So the question is, what do we mean by metabolism, right? So this is referring to all of the chemical reactions which take place within an organism, and it helps that organism to maintain life. And all of these chemical reactions can be divided into two categories. The first category is catabolism, where the metabolic reactions are involved in breaking down molecules to extract energy. An example of a molecule which is broken down to extract energy is glycogen. So glycogen is a polymer of glucose. So notice that there are a number of these glucose subunits which are linked one to another. So that is what glycogen is, and when you exercise, the glycogen in your muscles can be broken down to make glucose, and that glucose can be used to make ATP. Now the second type of reaction is anabolism, where these are reactions to synthesize building blocks to produce new molecules, and the joining of glucose molecules to make glycogen is an example of anabolic reaction. And the goal for us in this lecture is to mainly focus on the catabolic reactions to see how ATP can be made. So let's start our discussion by thinking about how plants can make carbohydrates by using energy from the sun. So in the process of photosynthesis, plants are going to use the carbon dioxide from the atmosphere, and it's going to combine it with the water from the ground, and the sun's energy will be used to bring these carbon-carbon atoms together and eventually make carbohydrates. And also oxygen is going to be released in the process. Now in order to make carbohydrates like this, it's going to require a lot of energy, because bringing those two carbon-carbon atoms in close proximity and linking them will require energy, right? So we see that there is this chemical energy which is present between these carbon-carbon bonds. And when we eat a meal, whether it is plant food or animal matter, our body is going to extract the energy from the food and convert it into a form which we can use. And the form which we can use is going to be ATP, also known as adenosine triphosphate. So here's a box indicating that an adenosine molecule is linked to a ribose sugar, and that's linked to 3 phosphates. And notice that adding these phosphates to this adenosine is going to require energy, right? And we see that to add the first phosphate, there's a little bit of energy required. To add the second phosphate, you need a lot more energy, but adding this third phosphate is going to need even more energy. And the reason for this is that the phosphates have negative charges, and like charges will repel, and that is why there is energy required in bringing these phosphates together and linking them. Now whatever energy our body is going to use to bring these phosphates together, that is the energy which will be used to fuel various cellular activities. So the chemical energy within these phosphate bonds can be used to do mechanical work, like the movement of muscles, for example. So here's a real life analogy. Like when you try to shoot an arrow from a bow, the energy you put into drawing back the bowstring is what will power the arrow being shot forward. So similarly, we see the energy in these phosphates bonds will what be used to fuel cellular activity. So here's an overview of what happens when glucose is going to be broken down. So notice that the glucose has got 6 carbon atoms. Each of those carbon atoms will be converted to carbon dioxide, and water will be produced as a side product as well. But notice that in the series of steps which are involved, there will be the release of high energy electrons, shown here by this red circles with a E in the middle, and there will also be these protons or hydrogen ions which are going to be released as well. Now these high energy electrons and protons, they will be carried by these electron carriers, right? And the two types of electron carriers we consider are NAD plus and FAD. The full meanings of the abbreviations you don't need to know, but just know that these are derived from vitamins. So that's why vitamins are necessary in our diet. And NAD is made from vitamin B3, and FAD is made from vitamin B2. So once they carry the electrons, they get converted into this NADH and FADH2. And these high energy electrons, they will be used in steps further downstream, which will eventually be used to make ATP. But notice that these high energy electrons, they are going to combine with oxygen to eventually form water. And so at a very high level, we see that for the complete breakdown of glucose, there will be around 30 different steps, and all you see here is 2 arrows, but there are actually 30 different steps, and there will be around 30 molecules of ATP produced per glucose molecule. And when a glucose molecule is completely oxidized, it yields carbon dioxide, water, and also ATP. So considering this overview, let's talk about what happens in the breakdown of carbohydrates. So in the breakdown of carbohydrates, we find that there will be 4 major pathways or steps we consider. The first step is glycolysis, right? So in glycolysis, notice that one glucose molecule is made into 2 pyruvates, and this is a step which takes place in the cytosol, right? This is the only step which takes place in the cytosol. The rest of it takes place in the mitochondria. So the pyruvate is going to be converted into acetyl-CoA, and the acetyl-CoA is going to enter the citric acid cycle, and the citric acid cycle will yield more high energy electrons, which will be transported to the electron transport chain, and thereby ATP can be made by transporting these electrons. So let's go into each of these steps in a little more detail. So what does the term glycolysis mean? So glycolysis is referring to sugar or glucose splitting. So notice that in glucose there are 6 carbon atoms, and it gives rise to 2 pyruvates, right? And so each pyruvate has only 3 carbon atoms. And let me just mention that as we go through these various pathways and steps, I need you to know only a couple of things. Know what goes into glycolysis and what is produced as an outcome, and also the number of carbon atoms. Those are the only 2 things I want you to keep in mind. The other components which are produced in between and their structures, you don't need to worry about it in this course. If you take a more advanced biochemistry course, you will learn about all those structures in detail. Notice that there is also no oxygen required in glycolysis, and this takes place in the cytosol, as I mentioned previously. And early on in glycolysis, you have 2 ATP molecules being used up. So this is like priming the pump. You invest 2 ATP molecules, but at the end of glycolysis, you end up with 4 ATP. So this means the net yield of ATP is only 2, because 4 minus 2 is 2. And in the meanwhile, there are these high energy electrons which are being carried by NAD, and that will be taken to the electron transport chain. So up till this point, we see that glycolysis does not yield too much ATP. I told you that from a single glucose we can make up to 30 ATP, but right now we only have a net yield of 2 ATP. So how is more ATP produced? So we will consider what happens to pyruvate. Now pyruvate can have 2 fates. It can either be made into lactate or into acetyl-CoA. So whether it's turned into lactate or acetyl-CoA depends on the amount of oxygen available. So if there isn't enough oxygen available, then we find that the pyruvate will be turned into lactate. And lactate, it's being produced in the muscle when you are exercising, and that can be sent into the bloodstream to the liver, and in the liver it can be turned back into pyruvate, and the pyruvate can be made back into glucose, and the glucose can be sent back to the muscle. So we see that gluconeogenesis takes place in the liver, but notice that a lot of ATP is being involved in making the glucose. So the question is, why would the body choose to make this much why would it choose to use as much ATP to make glucose when you only get a net yield of 4 ATP from a single glucose molecule? So the question is why, and the answer, one word answer, is speed, right? So the reason the body is choosing to make glucose in the liver is that the ATP can be produced very quickly through glycolysis. Now if it were to use the other pathway of making it into acetyl CoA, we can make a lot more ATP from a single glucose molecule, but it won't be produced as quickly. So here's a real life analogy. All of us here are currently here in Fort Collins, and let's say you need to go catch a flight in BIA, right? And you get an Uber to take you down, and you realize that there's a lot of traffic, and so you tell the driver, I'm going to pay you double if you get me to the airport in time. And then by the time you reach I-25, you see that it's a really long line of cars in traffic, and so you say, I don't care how much it costs, get me to the airport in time and I'll pay you 4 times what the meter shows, right? So this is an analogy of how you are not caring about how much you spend as long as you get where you need to be on time. So the body is going to use this anaerobic pathway to make the ATP very quickly, because the body is trying to let's say get away from a danger, right? So that's why the body is going to use this less energy efficient way to make the ATP pretty quickly. And so just to repeat what I said, when limited oxygen is available, pyruvate is shunted to form lactate. Now on the other hand, when oxygen is readily available, the pyruvate is converted into acetyl CoA. So keep in mind that there were 2 pyruvates produced from one glucose molecule, and so you can have 2 acetyl CoA being generated. So among the 3 carbons in the pyruvate, one of those carbons is going to be converted into carbon dioxide, whereas the other 2, they're going to be linked to coenzyme A to form acetyl CoA. And so coenzyme A is also a derivative of a B vitamin, and this is derived from vitamin B5, and this is just for your curiosity. I'm not going to tell you what vitamin is used to make NADH, what vitamins used to make FADH2. This is just for your curiosity's sake. And notice that this is an irreversible step. Once pyruvate is converted into acetyl CoA, there's no going back. The acetyl CoA has to enter into the citric acid cycle in the mitochondria. So now let's talk about the citric acid cycle. So before we go through the various steps in the citric acid cycle, I just want to focus on what the name means. So let's focus on what we mean by cycle. So in the very first step, that is oxaloacetate being one of the reactants, right? And in the very last step, oxaloacetate is going to be regenerated again. So that's why it is a cycle. And in the very first step, notice that oxaloacetate, which has 4 carbons, is going to combine with the 2 carbons from the acetyl CoA, and we see that citric acid is produced in the very first step, and that's the reason why this is called the citric acid cycle. So the 2 carbons which were linked to oxaloacetate to make citric acid cycle, 2 of those carbons will be lost as carbon dioxide, right? And so we see that pretty much all of the carbons which were in the glucose molecule, they have been converted into carbon dioxide. And I hope this explains why our cells produce carbon dioxide, right? When we breathe in oxygen, why does it turn into carbon dioxide? It's because of the citric acid cycle and the conversion of pyruvate into acetyl CoA. Is that clear, folks? Why we produce carbon dioxide? Okay. Moving on. Now let's see what are some of the reactions which take place in the citric acid cycle. So there is one GTP molecule being produced in the citric acid cycle, and this GTP is structurally similar to ATP. So as far as we are concerned, we can just say that it's used to make one ATP molecule. So for every citric acid cycle, you have one ATP molecule being produced. And we also see that there are a lot of high energy electrons which are released. So we see that there will be 3 NADH molecules which carry the high energy electrons, and 1 FADH molecule carrying the high energy electrons. And we find that oxaloacetate is going to be regenerated in the final step. And because there were two pyruvates produced from a single glucose molecule, there will be two acetyl-CoA produced as well, and so for every glucose molecule the entire citric acid cycle will repeat twice. And so we see that there's a lot of high energy electrons which are going to be carried from the citric acid cycle all the way to the electron transport chain. So in the electron transport chain, we find that the high energy electron carriers will finally be able to give up their cargo, and as the electrons are being moved from one protein complex to the other in the electron transport chain, they're going to give up their energy, and the energy which is given up by the high energy electrons is what will actually fuel the enzyme which makes ATP. So as you can see in this cartoon here, the electron transport chain is taking place in the mitochondria, and let's just orient ourselves to see where in the mitochondria this electron transport chain is taking place. So the mitochondria has got two membranes. It's a double membrane structure similar to other organelles in the cell, like the nucleus also has a double membrane structure. And so there's the outer mitochondrial membrane shown here, and then here's the inner mitochondrial membrane, and it is in the inner mitochondrial membrane where you find the electron transport chain. So if you zoom in closer, here's the cytosol shown here in yellow, here's the outer mitochondrial membrane, and then here's the space between the outer mitochondrial membrane and the inner mitochondrial membrane. So notice here that the electron transport chain is just shown here as a white box, but there are actually four different protein complexes where these electrons will be transported. So as these electrons are transported, they will give up their energy, and these energy depleted electrons will be combined with the oxygen in order to make water. And so the hydrogen ions, the energy depleted electrons, and the water and the oxygen will combine to form water. And if oxygen is not available for this final step of the electron transport chain, this can actually halt the ATP synthesis. So I hope you finally understand why we need to breathe in oxygen. Without the oxygen being available for the final step of the electron transport chain, our body cannot make ATP, and that is the reason why a number of poisons will actually try to target the electron transport chain, so that if your body can make ATP, then the cell will die and eventually the person will die as well. But I still haven't answered the question, how is the energy from the electrons used to make ATP? I told you how oxygen is used, how water is produced, but I haven't told you how the high energy from the electrons is used to make ATP. So if you take a closer look at this figure here, notice that here is complex 1, complex 2, complex 3, complex 4. In each of these complexes, the electrons are being passed from one complex to the other, and the energy which is given up by the electrons is going to be used to move protons, or in other words the hydrogen ions, from the mitochondrial matrix into the inner membrane space. So the hydrogen ions are going to be moved into the space between the outer and the inner mitochondrial membrane. Now notice that as the electrons are being passed, and as more and more of these hydrogen ions are going to start accumulating in this inner membrane space, eventually there will be a electrochemical gradient formed. So not only do you have a higher concentration of hydrogen ions in this inner membrane space, but these hydrogen ions are also charged, and that's why you have this electrochemical gradient. And these hydrogen ions will want to flow down that concentration gradient, and they need a channel through which they should flow through, right? And that channel for flowing through is the ATP synthase enzyme. So this purple structure here, this is the ATP synthase enzyme, and this will permit a channel for the hydrogen ions to flow down the concentration gradient. And as it flows down the concentration gradient, it causes this ATP synthase enzyme to rotate, right? And as they rotate, there will be some pockets opening where the ADP, adenosine diphosphate, and the phosphates can enter in, and there they will be combined into ATP. So as this ATP synthase enzyme rotates, it's going to cause the ATP to be synthesized and it is released. So is this clear, folks, how the high energy electrons can eventually be used to make ATP? So the majority of the ATP synthesis comes from the high energy electrons, not the in between steps where ATP is made in glycolysis or in the TCA cycle. This is the main money maker, right, in terms of making ATP. So here's a quick think, pair, share activity, right? And these are two questions you should be able to answer based on what we have been talking about so long. So when you're jogging at a relaxing pace, why is it that you can go on for almost an hour, right? But if you are sprinting, you cannot sustain it for more than a few minutes. And I just want to qualify relaxing pace by saying that for some of you, a 7-minute mile may be a pretty relaxing pace, right? I've tried a 7-minute mile once in my life and I thought I would die, right? And this was a long time ago when I was still in grad school. But it really depends. So why do you think a high intensity activity cannot be sustained for too long, but relaxing activity can happen for a long time? Why? Yes? Alright, folks, let's bring it back together. Do you have any responses? What happens when you're increasing the intensity of an activity versus when it's happening at a lower intensity? Here's a shy group here. Not even making eye contact. Oh, yes. Okay. Okay, thank you. So just to repeat what you said, when you're engaging in a high intensity activity, you aren't getting enough oxygen, right? And that's why the pyruvate is not being converted acetyl CoA, and that's why you're getting less ATP, right? And so you're using up the substrates a lot quicker, and so you're not getting enough ATP. Whereas if you are at a more relaxing pace, you get more oxygen, and so you can get more ATP for the available amount of glucose in the body. So this brings us to a recap of what we just discussed, and this slide here, it's very important, right? I won't ask you to draw it out, but if you can draw this out from memory, it will really help with answering a number of questions, right? And so let me walk you through this slide, and we can also answer the question in the previous slide by going through this slide. So here's glucose. It's got 6 carbons, and after glycolysis you produce 2 pyruvates each, which has 3 carbon atoms. And notice that you have only 4 ATP being produced. There's not much ATP, and there's some NADH molecules carrying the high energy electrons. Now the pyruvate can be converted into lactic acid when there isn't sufficient oxygen, especially when you're engaged in a high intensity activity, and that lactic acid can be converted into pyruvate and back into glucose in the liver. Now all of this is taking place in the cytosol or in the cytoplasm, and there is no oxygen being used, so this is anaerobic metabolism. But if there is sufficient oxygen available, then the pyruvate will be moved into the mitochondria, where it is converted into acetyl-CoA. And notice that among the 3 carbons from pyruvate, 1 is lost as carbon dioxide, and only 2 carbons from the glucose molecule is linked to coenzyme A. And each of these carbons, they will enter through the citric acid cycle, which is also called the Krebs cycle. You can use either term interchangeably. And in the citric acid cycle, both those carbons will be lost as carbon dioxide. And notice that you only have less amount of ATP being produced in the citric acid cycle as well. However, the high energy electrons, they're going to be carried into the electron transport chain, where they will give up their energy to fuel the synthesis of ATP, and also these energy depleted electrons will combine with oxygen and with the hydrogen ions to eventually form water. So it is this anaerobic pathway which you use during a sprint, and that's why you make the ATP very quickly, but you don't make as much ATP. Whereas in the aerobic pathway, which would happen during a relaxing walk or a half marathon, that's the time you're going to have a lot more ATP being produced for a given glucose molecule, and that's how you can sustain it for a longer time. Is that clear, folks? When you're using the aerobic pathway, you're making more ATP for a given glucose molecule. So if you want to burn lots of calories, which one should you engage in a short amount of time? If you want to burn more calories in a short amount of time, sprinting, right? Okay, great. Is it easy to do? No, it's not. So moving on, extracting energy from fat, we find that the very first step involved is going to be the digestion of lipids using the lipase enzyme. So lipase will break the glycerol backbone away from the fatty acid chains. Now, how many carbons does glycerol have? 3. Glycerol it's a 3-carbon alcohol when we talked about the triglyceride structure. So glycerol has got 3 carbon atoms, and so with just few structural modifications, it can enter into the glycolytic pathway, and one glycerol can be converted into a single pyruvate. But we know that from a single pyruvate, you may not get as much ATP. But all of these fatty acid chains, they've got lots and lots of carbons. So those carbons, they're going to be taken 2 at a time through a process known as beta oxidation to eventually make acetyl-CoA. So the term beta oxidation, it's not there in your handouts, so please write that down. You know, the fatty acid chains will undergo beta oxidation to form acetyl-CoA. So let's consider what's happening in beta oxidation. So we see that this is going to take place inside the mitochondria, and this is a cartoon of a fatty acid chain. Notice that each of these blue circles, they're indicative of a carbon atom, and 2 by 2 these carbon atoms will be linked to coenzyme A, and then they will be converted into acetyl-CoA. And the reason they use the term beta oxidation is referring to the number or the term on the carbon, alpha or beta carbon. That's why they call it beta oxidation. And all of these acetyl-CoA will enter into the citric acid cycle, and it's going to generate lots of NADH and FADH2, which will eventually be passed on to the electron transport chain, and that's how lots of ATP is going to be made. So you see how once you understand the aerobic and anaerobic pathway, it's very easy to understand what's happening in the fat metabolism as well. So an important thing to consider is that for transporting the fatty acids into the mitochondria, there is this substance called as carnitine. And because carnitine is responsible for the transport of fatty acids into the mitochondria, it's been claimed that it can be a fat burner. So more carnitine in the body means more fat will be burned. This is really not got as much scientific support, right? So that's even evidence that it can lead to muscle weakness. But I think since some of you may be interested in taking carnitine supplements, just keep in mind that it's there are some health concerns, like it can cause muscle weakness and so on. So now we'll move on to the third type of macronutrient, making ATP from proteins. So we see that the body would preferentially use either carbohydrates or fats to make ATP, and it's only during times of starvation where the body would actually break down proteins for ATP synthesis. So as you can see, proteins are all made up of amino acids, and let's see what happens at the level of a single amino acid on how it is being processed. So the amino group in the amino acid cannot be actually metabolized by our body, and so it will be stripped off by a process known as deamination, and these amino groups will be excreted in the urine as urea. Whereas this remaining carbon skeleton, which includes the carboxylic group and the R group, which is linked to the alpha carbon, all of that can enter into different steps of the catabolic pathways. And here's an example of what I mean. For example, the structure of alanine is such that it can easily be converted into pyruvate, whereas the structure of methionine, for example, is such that it's going to enter somewhere in the middle of the citric acid cycle. So this means that if amino acids are entering different parts of the metabolic pathway, depending on where it enters in, it can produce a different amount of ATP. So what do you guys think? Do you think alanine will produce more ATP, or do you think methionine would produce more ATP? You can just go ahead and answer this poll everywhere question. Okay, we have a few responses. Most of you said alanine, and that is correct, right? So if you notice here, the actual number says that alanine can yield more than double the amount of ATP as compared to methionine, around 12.5 compared to 5. And the reason for this is if because it enters earlier on in the pathway, it can end up releasing more high energy electrons, which are carried by NADH and FADH2, whereas if it enters later in the pathway, you have fewer high energy electrons being carried by the electron carriers. So the bottom line is the complete breakdown of amino acids yields urea, carbon dioxide, water, and also ATP. And the reason for water being produced is because it's mainly using the aerobic metabolism to break down proteins. So now we move on to question number 2 of today's lecture. It is, how is metabolism related to respiration, right? And I want to introduce you to this term known as the respiratory quotient, or the RQ value, and this is an index of which nutrient is being used by the body to generate ATP. So just by looking at the RQ value, you can say whether proteins are being used, carbohydrates are being used, or if a combination of proteins and if a combination of carbohydrates and fats are being used. So the RQ value is defined as the volume of carbon dioxide being exhaled divided by the volume of the oxygen being used. So you could determine a person's RQ value in a lab setting by putting a mask on them and seeing what RQ value they are producing. And for even different types of foods, you can determine what would be the RQ value if you ate a specific type of food, right? So what they would do is they would take proteins, carbohydrates, fats, and burn it in a lab setting and calculate how much oxygen was used to burn this completely and how much carbon dioxide did it release, right? So these three values I would hold you responsible for, right? Because by knowing what is the RQ value for carbohydrates, proteins, and fats, you can actually indicate what type of an activity a person may be engaging in, right? Or what type of fuel the body is using to make ATP. So for those of you who are curious, I've included this one slide which shows you how these reactions took place, right? And how much oxygen was used and how much carbon dioxide was released, and that's how they came up with these values 1, 0.7 and 0.8. And this is beyond the scope of our discussion, and that's why it's not in your handouts. But if any of you are curious, email me and I'll send you this slide. So what I want you to realize is, why is the RQ value for carbohydrates higher than the RQ value for proteins and fats? I repeat, I need you to know why the RQ value for carbohydrates is much higher compared to the RQ value for proteins and fats. And this is something you should be able to explain based on what we've been talking in today's lecture. So keep in mind that when it comes to breakdown of glucose and glycogen, it can use both the aerobic pathway and the anaerobic pathway, right? If you need to make the ATP very quickly, like during a sprint, the anaerobic metabolism can be used. And so if anaerobic metabolism is used, it means you're going to use less oxygen. And if you use less oxygen, that means the denominator in the RQ value is going to decrease. And if the denominator decreases, the RQ value will go up. Is that clear? So because carbohydrates uses the anaerobic pathway, there will be less oxygen used, and as a result the RQ value will increase. On the flip side, we see that the breakdown of proteins and fats is mainly going to use the aerobic pathway. And because it's using, I know that for fat breakdown there's a small amount of glycogen which is produced, which can enter the maybe the anaerobic pathway, but most of the fat is broken down using the aerobic pathway, and similarly even most of proteins are broken down using the aerobic pathway, and that is the reason why it requires more oxygen. So as the denominator gets larger, the RQ value will get smaller. Is that clear, folks? So I hope you're just not memorizing the three values 1.0, 0.8, 0.7, but you know why carbohydrates have a higher RQ value than the proteins and fats. So this is a slide which we are going to revisit on Wednesday, right, where we see how the RQ value shown here on the y-axis is going to change with respect to time, and you also see that the RQ value will change based on whether you're using carbohydrates or fats at rest, and then we'll see what happens when you're using creatine phosphate. I haven't introduced you to that as yet. And then what happens when you have anaerobic activity, it goes up and then eventually it starts decreasing. So we'll see how the RQ value can change with time depending on what fuel the body is using to make ATP. Alright, so this is something we'll revisit in more detail when we come back on Wednesday. But for now, let's just quickly go through some of the poll everywhere questions which are at the end of your lecture, right? So this is the question which we worked out in class on Friday. Hydrogen bonding between the carbonyl oxygen of one amino acid and the hydrogen on the amine of a nearby amino acid is responsible for the blank structure in proteins. Is it primary, secondary, tertiary, or alpha helical, or more than one answer is correct? We got a lot of responses already. Most of you said more than one answer is correct, and I'm glad that most of you said more than one answer is correct because we did this question on Friday, right? So what are the right answers? Secondary and alpha helix, right? And as shown in the slide here, we see that secondary structures include alpha helices and beta sheets, and if, let's say, if there was beta sheet here, that would have been the right answer as well. Okay, next question. In the lipid mosaic model of the membrane, of the cell membrane, blank normally forms the ion channels. So do you think it is the amphipathic phosphate head groups, or is it the alpha helices, or would it be beta sheets, phospholipids, or triglycerides? Which of the following structures would be forming ion channels? Is it? Great. Okay, so most of you said alpha helices, and let me just walk you through this because I feel that there are a number of you who have said something else. Right? So the correct answer is alpha helices. So the option A said amphipathic phosphate head groups. If answer A was the option, you don't need any ion channels because just the plain lipid bilayer is made up of amphipathic phosphate head groups, right? So this lipid bilayer has got these phospholipids in it, and you don't have proteins channels made of just these phosphate head groups. Instead, you have it made of these alpha helices, and this is a concept which we will be revisiting in lecture 7, and we see that the channels, the transmembrane proteins, are mostly made up of these alpha helices, and the reason for that is that there will be maximal number of hydrogen bonding between the amino acids in the alpha helix, and secondly, the length of alpha helix is around 20 amino acids, and if you think about the distance between each amino acid, around 1.5 angstroms, that length is sufficient to span the width of the lipid bilayer, and so the ion channels are mostly made up of alpha helical structures. Beta sheets, I know there are beta barrels, but this is not as common, and phospholipids is just referring to the lipid bilayer, and triglycerides will not permit something which is hydrophilic to get through. Alright, so with that, we will stop class here because I want to be in sync with both sections, and I'll see you all on Wednesday. And folks, please try to answer the polar viewer questions at home, so that when you come here, it's just a review of your knowledge, right? So thank you all for your time, and I'll see you on Wednesday.